M is for the way meiosis occurs. E is for the equator where the chromosomes line up. I is for interphase before meiosis. O is for opposite poles of a cell. Bum 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 bum. S is not for somatic cells. I is for biointellectuals. S is for division of sex cells, and all of this spells meiosis. Hey guys, it's Chloe and Sophia, and today we're going to be covering the process of meiosis.
Meiosis is a process in which gametes, also known as sex cells, are dividing. Meiosis starts out with one diploid cell and results with 4 haploid cells.
Before meiosis can even begin, the cell grows and duplicates DNA. This is called interphase. Interphase consists of 3 parts, G1, S, and G2. In G1, the cell begins to grow in size by taking in water and by making more proteins. In the S stage, DNA is duplicated so that the DNA can be evenly split into each new cell. In G2, organelles are duplicated so the cell will be ready to divide. The cell also continues to grow.
Let's talk about prophase 1. Prophase 1 begins with the condensing of genetic material or chromatin into chromosomes. Spindle fibers are also formed during this time. The chromosomes then connect in homologous pairs where crossing over occurs. When the genes cross over, there is a recombination of genes on the new chromosome. This allows for genetic variation. As all of these things are happening, the nuclear membrane disappears.
The second phase is metaphase 1. During metaphase 1, the homologous pairs of chromosomes are pulled in the center of the cell by spindle fibers.
Once the spindle fibers attach to chromosomes, anaphase 1 can start. During anaphase 1, the spindle fibers pull the homologous chromosomes apart to separate sides of the cell.
Now for the final phase of the first part of meiosis, telophase 1. During telophase 1, a cleavage furrow forms and cytokinesis splits the cell into 2. A new nuclear envelope is formed in each of the recently created cells.
Wait, but that's not all. Meiosis goes through a second similar division as the first. The second division is pretty similar to the first, except the 2 cells are splitting rather than 1. The second part of meiosis starts with prophase 2. During prophase 2, the spindle fibers reform and separate, and the nuclear envelope disappears.
In metaphase 2, single chromosomes line up on the metaphase plate and prepare to be pulled apart in the next phase. How is this different from metaphase 1? In metaphase 1, the chromosomes line up in the center in homologous pairs. Meanwhile, in metaphase 2, the chromosomes line up as single chromosomes.
In anaphase 2, the sister chromatids are pulled apart by the spindle fibers to opposite poles of the cell.
In the final step of meiosis, a few different events occur. A cleavage furrow develops so cytokinesis can take place, and the 2 cells divide into 4 haploid daughter cells. The daughter cells' nuclear envelopes and membranes become completely formed, and meiosis is officially over. N equals 2 because there are 2 sets of chromosomes.